Hello, and thank you for joining us for the latest webinar from DuPont Tyvek Medical and Pharmaceutical Packaging. Today's session is co-sponsored with WESTPAK and Eagle Medical. We know your schedules are all busy, so we'll get right to it. My name is Jen Benolken, and I am an MDM and regulatory specialist with the Tyvek Medical and Pharmaceutical Packaging Group in North America. I'll be moderating today's session. Prior to jumping into the discussion, though, I wanted to share a few pieces of housekeeping. First, the presentation materials are owned by and copyrighted by DuPont Tyvek Medical and Pharmaceutical Packaging and are co-sponsors for today's sessions. The individual speakers are solely responsible for their own content and opinions. Slides will be available to attendees. Please check your handout section in the control panel. Second, if at any time you're experiencing troubles hearing the discussion, please check your equipment, unplug, plug it back in, and or disconnect any other devices that may be using streaming services on your network. If all else fails, just try logging off and back in. Due to the large number of attendees, everyone will be muted. Third and last, regarding questions, we fully expect and welcome questions throughout the webinar. Because we have so many people on the line, we have set it up to have you submit your questions in the questions window in the control panel. We will address questions during the panel session at the end of the program. We may not be able to get to all questions during the session, but we'll make sure to follow up with you later if needed. We will also share additional information via our LinkedIn channel. If you haven't followed us yet, please do so. The genesis of today's webinar started at a conference over a year ago where a few of us discussed hosting an in-person session for the startup community in the Bay Area at the WESTPAK facility. It was planned for last May, but we're all too familiar with why that didn't happen. Nevertheless, we persevered and continued to talk and evolved into this webinar session. We think the information contained within this is relevant and timely to our entire community, not just the startup community. Our theme is balancing data with risk to improve patient safety. While each of these topics is vastly different from one to the next, each of them are focused on how we can use what we know, the data, and what we don't know, the risk, to improve our patients' outcomes. Patient outcomes has been what's kept me involved in this industry for almost 30 years. For while most of us equate the patient to some unknown person, the fact is it could be your coworker, your mom or dad, your child, or you. With that, let's dive right into our first presenter, Dan Floyd. He's going to be joining me here via the camera in a second. As he's joining, I want to say I don't want to waste your time by reading the individual presenters' bios. They're on the original sign-in sheet when you first signed up. And because they're not too nervous enough, they're not nervous enough already being presenters for today's event, I am going to tell you the skinny on each presenter. So Dan, I'm going to fry you here. So Dan is into deep sea diving. Next time you run into him, make sure you ask him about his infamous trip to the Turks and Caicos, the trip that wasn't. Dan, it's all you.
Right on. Hey, hello and welcome. I'm Dan Floyd with DuPont, and just wanted to thank you guys for joining today. Our theme today is patient safety, and as such, what we want to do is talk about a lot of different topics. One of the ones I'm going to talk about today is a lot of the issues that are going on in the sterilization industry. There's lots of challenges that are going on, so nothing, I guess, screams patient safety if there's some challenges that are affecting about 90% of how all the medical devices that are sterilized. So that's what we'll talk about today. There's a lot of issues going on with ethylene oxide. I think a lot of people have seen some of that in the news. There's also some issues with gamma sterilization. And again, these two methods make up more than 90% of large-scale industrial sterilization of medical devices. So definitely a patient safety issue. We'll talk about some of the new emerging modalities. Can any of these replace ethylene oxide? We'll talk about that. And then some of the trends on what people are doing to deal with this issue. So what happened, just going through some of the key dates, ethylene oxide sterilization really never changed much in the last several decades. It's been pretty constant. And then 2016, the big deal was that the US EPA classified ethylene oxide from a suspected carcinogen to a known carcinogen. Now the data from that wasn't conclusive, so they do admit that it's short of conclusive. There's some animal studies, but the human studies didn't really show a definitive link. But even so, that's been classified as a carcinogenic to humans now. So that was kind of what set the ball rolling. So in 2018, the EPA's National Air Toxics Assessment, basically looks at air monitoring all across the U.S. and correlated it with some higher, what looked like what was potential higher areas of cancer. Some of these areas correlated with contract sterilization facilities, although a lot of these are in industrial areas. We'll talk about, there's a lot of other sources of ethylene oxide, but that's kind of what started this whole issue. And then February 2019 was what really kind of shook the industry up. The Illinois State EPA, what started somewhat as a political dispute between the the governor and the former governor, and there's some issues involved with that, but they shut down the Sterigenics Willowbrook EO facility there, which really had a huge impact on the industry. That facility alone dealt with 510 medical device types and over 120 medical device companies that were affected by that. And as many of you know, to validate a new facility, to move from there in outside of Chicago, Willowbrook, to wherever you're going to move to, say you're going to move to California, that is something that's going to take months to do, tons of validation work. So it's not an easy or a quick process to do. So it created tons of fears of device shortages, that this could really affect the medical device industry. So later that year, in August through October, that issue sort of moved down towards the Atlanta area. There's 4 different facilities that were affected there. The Sterigenics facility in Georgia, 2 Becton Dickinson locations, and another contract sterilization facility, it's called Sterilization Services of Georgia. They were all affected by this. Many of these shut down for a period of time. And, you can imagine all the potential issues with shortages now that we're talking 5 to 6 different facilities being affected by that. So there really were fears that this was going to happen in many locations all over the country. But then March 2020, everybody knows the pandemic kind of hit full force, and there was huge shortages of PPE. Many of you have seen on the news where first responders and nurses, doctors were even forced to reuse surgical masks, re-sterilize those, reprocess those, just because there were a shortage of it. So the FDA urged the state of Georgia to allow the Sterigenics facility to reopen due to a lot of the critical products that were being processed. And in September 2020, these new expected EPA emission limits were expected to come out originally early 2020, but they kept getting pushed back, obviously, because of the pandemic. And then in September 2020, they've now delayed the release until sometime probably in the next month or 2. They're expected to come out with these new limits, which are obviously going to affect the industry greatly. And there's a lot of controversy about what those should be, how the data was assessed, and the assessments that were done to determine that. So we'll talk a little bit about that. So when you talk about potential patient safety, the numbers are just enormous. There's over 2 million different medical devices listed in FDA's Global Universal Device ID database, and 40% of all those devices are sterilized. And of all the devices that are sterilized, 50% of those are sterilized with ethylene oxide, 40% are sterilized with gamma. Now this is wide scale, real large sterilization, not what's done in hospitals. Hospital sterilization is a lot different, but you're talking 90% of all devices that are sterilized on a large scale, are affected by some of the issues that we're going to talk about. So if EO sterilization was to be banned, and they're not necessarily talking about banning it outright, but if this issue were to spread over large areas, 20% of all medical devices, since 40% of all medical devices are sterilized, that's 20% of all of them would be at risk and no longer being available to patients. And that's, the number of that is just enormous. We're talking 20 billion devices just in the US every year are sterilized with ethylene oxide, 20 billion devices. So, and many of these devices can't be sterilized with alternative methods. We'll talk a little bit about some of the methods that were proposed. Really, there's 3 main reasons why there's not another type of sterilization that can just come in and replace ethylene oxide. One is scalability. Some of these EO sterilizers can sterilize up to 30 pallets at a time. You're talking a tractor trailer, huge truckload of product in a chamber. And so just enormous scale. Availability, these contract sterilization facilities are all over the US. And the second reason is the inability of these other sterilants to be able to penetrate into some of the hard to sterilize parts of the device or the product load itself. When you box it up, put it on a pallet, in some cases maybe it's shrink wrapped. It's just hard for some of these to penetrate as good as ethylene oxide does. Ethylene oxide is a very small molecule, so it penetrates very well. That's really it's probably its biggest advantage, is its ability to penetrate through polymers and through a lot of the different plastics. And then finally, if you look at some of the other main ways to sterilize devices on a large scale, radiation, there's a lot of different material compatibility issues with many devices. So the kits, the custom kit business, you might have 40 components, 50 components. Some of those probably can't be sterilized any other way. So then the entire kit has to be sterilized with ethylene oxide. So the numbers are just huge. So when you look at that, it's the US EPA's role in this is under the Clean Air Act. They set limits on different air pollutants, and ethylene oxide is considered an air pollutant. So they did a risk assessment, which was called IRIS, Integrated Risk Assessment, and they looked at the risk of cancer, things like that, and what these emission limits should be. And many in the industry feel that they use flawed science on how they came up with this. There's an alternative model that the state of Texas came up with, the Texas Commission on Environmental Quality. They based it, both EPA and the Texas Commission on Environmental Quality, they looked at the same study that looked at 17,500 workers in the sterilization industry. And of that, they found 53 cases of lymphoid cancer. But if you look at the EPA's model, it way over predicted the amount of cases that you would expect from their model, almost more than double of what it was, almost triple of what the actual observed cases were. The Texas model actually is pretty much right on. It had 59 predicted cases, and there was actually 53, so it's much closer. The EPA risk range also is just several orders of magnitude lower of what they're saying the amount of EO that would cause cancer, several orders of magnitude lower than what is naturally produced in the body just through normal metabolic processes, which is, saying that what your body produces normally is, head and shoulders above what would cause cancer. So there's an issue there. And it doesn't take into account that the workers were exposed to much higher levels back in the day, back decades ago. I started in the industry in 1992 and working in several different contract sterilization facilities. There was no respirators. The operators would drive into the chambers, unload the chambers right after the cycle. The OSHA limits were much higher than they are today, decades ago. So now we have PPE, the practices are better, there's lower OSHA limits. So really the risk is less than it would have been back then. So here's a couple slides that I took from this Texas Commission on Environmental Quality. This is the data shows, this is the normal amount, this black line here shows the normal amount of ethylene oxide produced by your body, just normally through metabolic processes, through the oxidation of ethylene. And if you look at the EPA's maximum acceptable EO dose, it's way down here, way below the even 1 percentile, 40 times lower than even the first percentile of that normal amount. So you can see there's some issues with what's being proposed. In addition, they looked at ethylene oxide monitoring, background monitoring in different locations around the country. And they found even in sites that were nowhere near any contract sterilization facilities, there was 10 to 21 times the EPA's maximum acceptable range here of 0.0185 micrograms per cubic meter. So, it's kind of crazy that these limits would be lower than what's just normally in the background, miles and miles and miles away from these facilities. So AdvaMed came out with this data here. This AdvaMed is basically kind of a lobbying group on behalf of the medical device industry. And they looked at the amount of EO that's produced from several different sources. You look at a school bus, it can be 1 million times the amount of ethylene oxide emitted than the EPA's benchmark. If you have a charcoal grill, that would be 36 million times the amount of ethylene oxide, or even a gas generator, all the way up to 117 million times the EPA's benchmark that they're looking at. So just, normal car exhaust, truck exhaust is just, crazy amounts higher than what that would be. So the FDA came in, so that EPA is more dealing with the air pollutants and that type of thing, the environment. FDA obviously is looking at the medical device supply chain and patient safety. So they issued a challenge to the entire industry. And it was a two-part challenge. So the first one is said, okay, can we identify new sterilization methods and technologies? So is there anything out there that can replace ethylene oxide? So 24 different companies submitted ideas. And not every company that has a different type of sterilization submitted that. There's some out there that are good methods of sterilization that weren't submitted, but the FDA chose the following ones. And these are all great methods of sterilization, really nothing that's brand new. These have been out there for several years. You know, a lot of people are familiar with vaporized hydrogen peroxide, used very widely in hospitals. Nitrogen dioxide is kind of a newer one, supercritical CO2. These are all great methods of sterilization, and they all have great applications, but pretty much they're a niche-based type one. Now, accelerator-based radiation, that's probably the most promising. You have E-beam, and then X-ray, which X-ray has been around for a long time, but not really used on a large scale, mostly due to it's not very efficient. It's similar to E-beam, but what you do is you basically put like a tantalum filter, if you will, and slow down the electrons, and that releases X-rays. But because X-ray can handle such a large volume of product, they can do full pallets at a time. You're seeing X-ray facilities popping up all over. All the major contract sterilizers are building multiple facilities. So you'll see a lot of X-ray probably coming on. That's probably the most promising, to be able to take on a lot of different products that are currently sterilized with EO, if it can stand the material compatibility issues. So these were the ones that were looked at. Really, again, if you look at these, there's not one item here that could just plug in and totally replace ethylene oxide due to those three factors that I mentioned earlier. So the second part of that challenge was, okay, what can we do to reduce ethylene oxide emissions? And so if you look at this list here, many of these are dealing with looking at the EO process, optimizing it, enhancing it. In a lot of cases, what they're doing is minimizing the amount of gas that's used, cutting the gas concentration in half. If you cut that in half, then immediately you have just half the amount of emissions there alone. Other people are looking at only running it with a full chamber, so you're really maximizing the ethylene oxide that you are using. So again, this was a big US issue, but it's also kind of spread to Europe now. So Europe is assessing ethylene oxide. In mid-December, the EU Biocidal Products Committee, they adopted a negative opinion on EO. So they're looking at things, okay, this is a risk to health. So, they're going to do a negative opinion on that. But if there's nothing else that can be used, they have what's called a derogation, or really it's just an exception to that rule. And so basically, if you see here the text in red, that not approving the active substance, in this case ethylene oxide, it would have a disproportionate negative impact on society when you look at the risk to the public. And so this is ongoing right now. So they're really trying to get this exception, and most likely will, because even in this final report here that you can find online, this Biocidal Products Committee report on that, in their summary, they basically state in there that there is nothing that can replace ethylene oxide at this time. So most likely they'll get an exception. Europe will still be able to use ethylene oxide. That'll be valid for 5 years, and then that can be renewed on an ongoing basis, assuming that they can justify that there is nothing else that can replace it. Maybe in the future something will come up. So that's the issue with the US and Europe on ethylene oxide. So gamma also has some issues. There's been, not as critical as the ethylene oxide issues, but there's been definitely the two biggest issues you have with gamma sterilization is both over security and then the supply. So there's an interesting article, you can Google this article here. There was a truck that was carrying radioactive cobalt. So with gamma, they take cobalt 59, put it into a nuclear reactor, and it sits in there for 2 years, and then it comes out as cobalt 60, which is radioactive. That's what's used for the gamma knife procedure, for cancer treatments, what's used for medical device sterilization. But in 2013, some thieves stole a truck that was carrying this radioactive cobalt, and they didn't necessarily know what it was. probably thought it was something of value, maybe TVs or something that they could sell and make money off of. But this really spiked a lot of fears of terrorism. And, obviously after 9/11, that's always a big concern. So, they thought, well, could there be a dirty bomb? They could take this radioactive material and do something that. So there's been a lot of calls from different governments to phase out cobalt-60 in favor of things like E-beam and X-ray. In fact, there was in 2015, there was in Congress, the U.S. Congress, there was legislation that was proposed to phase out cobalt-60. Now it failed, but there is a lot of calls for that, so we'll have to see where that goes. The second issue, there's an article in the Wall Street Journal, will Tesla die for lack of cobalt? So the cobalt supply, because of the enormous growth of electric vehicles and batteries, this has been a big issue. 2/3 of all the cobalt that they use for these applications comes from Congo, which is an unstable country. There's issues with child labor. And so, having all those eggs in that one basket is a concern as well. And so of the cobalt-60 that they use, 80% of that's made in Canada and Russia. Canada used to have an old nuclear reactor that they had been making this, and they shut that reactor down and upgraded it to a new process. So there was a supply issue for a while, a couple of years ago, but then in March 2019, this new process, this light water nuclear reactor process was developed, and the first harvest of medical cobalt-60 was back up online in March of 2019, so that seems to be up and going. But this supply is always going to be an issue, especially with the enormous growth of electric vehicles, batteries, and things like that. So what's the industry doing? How are we dealing with this issue? Again, there's really not a replacement that's viable that you can just come in and say, okay, switch over to this method. So what a lot of companies are doing, this is a white paper that Steris has that I found on their website. They're doing something called sustainable EO validations. Sterigenics has a similar program, and most contract sterilizers are doing this. Most medical device companies are doing some type of work to optimize their current processes. So really what this involves in reducing the gas concentration. Historically, when I was doing EO validations for 25 years, we would always recommend, around 600 to 650 milligrams per liter. That's being cut in half now. People are looking at doing it all the way down to 300 milligrams per liter. And they showed Steris did a white paper that showed when they did that, they reduced that concentration all the way down. Then they were able to show the residuals on the product were also reduced in some cases by greater than 50%. So there's a big benefit there. Not only are you using half the EO, which the emissions are going to be a lot less, but then you have a lot less fugitive emissions coming off the product because the amount that's still on the product is a lot less than it started with. Again, Sterigenics has a process called reduce, reuse, reclaim. Steris is called sustainable EO. So a lot of different other contract sterilizers have that same process. So what are companies doing? There's again, the FDA came in and they're looking at, what can you do to your current process to help with this issue? So again, ethylene oxide is a gas. It has to penetrate into the product, and then it has to be able to come out of the product after the cycle. So anything that can help penetration and enhance aeration is going to be critical. So FDA came out with a statement at the end of 2019. One of the items that they talked about is as soon as possible to reduce the amount of paper. So you have paper packaging, you have paper labels, you have instructions for use manuals, and in some cases these are almost like a phone book. They're in multiple languages, and multiply that over 30 pallets of every product having one of these booklets in there. Then that's a large amount of ethylene oxide. So paper, a lot of times what it'll do, it'll absorb EO, and so in some cases then it's going to require to have more ethylene oxide it's going to absorb more of that, so there's less available to the product, and you have to use more ethylene oxide. So that was one of the things that the FDA looked at to make that change. So finally, again, when we looked at that innovation challenge, that second part of the FDA, the summary of that is really the bulk of this and why it's a patient safety issue. There's really no viable plug-in replacement that you could do today. And this includes all of the innovation challenge winners. You look at a lot of those gaseous methods, they're typically smaller, they're not available as widely, And the scalability, you can't do 30 pallets in multiple locations. And so that's a big issue. The penetration is another one. And you look at things like E-beam and X-ray can have material compatibility issues. Steam obviously is too hot for a lot of the plastics. So there's really nothing out there today that's going to be able to just replace ethylene oxide as it is. So we're going to have to monitor this. The industry is moving to minimize the amount of ethylene oxides that's used, changing in some cases the way that the validations are doing. So you're not doing this huge amount of overkill, which maybe gave you an 8-hour cycle, years ago when you validated, but maybe that cycle really only needs to be an hour and a half or an hour. A lot of companies just didn't take the time to do the work to optimize that. So it was really faster just to pick a time. I'm going to pick 4 hours. Yep, 4 hours worked, and I can go on and get this validation done and put it behind me. But they didn't go back to say, hey, maybe we could have done this in 1 hour, or maybe we could have cut the gas concentration in half. So you'll see changes to how these validations are done, and maybe people are optimizing, only sterilizing full loads, where a lot of cases people would do minimum loads. And so, really trying to make it as efficient a process as possible. So again, that's where we're at with these two industry issues. We're going to basically keep monitoring that. EPA is expected to come out in the next month or two. Hopefully, we'll see what happens with that. Hopefully, it's a more practical approach than maybe what's currently proposed, and then we'll go from there. So thank you for your attention. I appreciate everybody joining, here live from the house. I wish we could have done this anywhere else, because I'm ready to go anywhere but in my house right now. So thank you, and enjoy the rest of the webinar.
Thanks, Dan. That was great. We really appreciate it. Next, we have Roy Morgan with Eagle Medical. I met Roy a few years ago through Jody Burks at that infamous seminar where we started talking about the concept of this webinar. So anyway, apparently, this is the question you need to ask Roy when you see him in person next, or maybe you just want to call him up. Roy's got a penchant for sports cars. Ask him about the Tour de France hill that he tried recently. Roy, it is all yours.
Hey, great. Thanks, Jen, and welcome everyone. Appreciate your time and wherever you might be, good morning, good afternoon, and good evening. Today, we're going to talk a little bit about sampling in relation to patient safety and risk. It's a very dense topic, so we're going to try and keep it snappy for you and get through some items that are difficult to understand. So I want to start off with one of my favorite quotes, because I'm sure for all the industrial experimentalists in the audience out there, you really appreciate what this says and how it's our constant battle to try and make sense out of the chaos that we're often confronted with. So let's take a look at an example for just a second. And I got to ask everybody, does this look like a mistake to you? Let's take a look. How's it? Are we able to do that? Okay, it looks like we're getting choppy video. There we go. This is really my favorite part right here. Ooh. So hopefully your experimental programs don't all end like this. And the question is, does that look like a mistake to you? Well, let's consider how it was framed up in terms of a key point that I want to focus on in the presentation, which is Deming's point number 8 about driving out fear. A lot of people in the industrial experimentation community tend to think that this is really part of the human relations part of building a business. And sure, it has something to do with that. I'm going to frame it up more in terms of mistakes and false positives and false negatives. Which are you more fearful of making? And which do you take more seriously as you develop a test program? Well, we all know, and for those that aren't that familiar, that there are 2 types of errors that can be made. We'll classify them in this case as producer's risk, a type 1, or consumer's risk, a type 2 error. Looking back at that video we just saw, Musk actually called it a successful ascent. They got all the data they needed, even though it ended in a fire disaster. So regardless of what you might be more fearful of making in your experimental career, a producer's risk error or a consumer's risk error, the punchline, no one in the organization is going to care, because as soon as that mistake is made, you're over budget and behind schedule. So let's look at how we can combat some of these things. That SpaceX mistake, that was really just an expensive sample. And we actually know exactly how much it cost. This is important, and we're going to talk more about it as we get through the talk here quickly. But it really identifies the crux of what the battle for perfect information is, right? It's this continual push and pull between fast, cheap, and great, as I like to call it. And the idea that we have in our experimental programs, through the selection of samples, the ability to get closer to great without giving up either of the other two. And we're going to look at that in some detail. We're going to talk through the stepwise analysis of how input changes to your sample selection, specifically in relation to an operational characteristic curve, can affect your patient risk. So let's take a look. All right, so how's it all calculated? I'm not going to put you to sleep with math today. We're going to deal with graphs, because that's always a little bit easier to comprehend. And in a short 20-minute talk about this topic, it's faster for everyone to grasp. But I always get asked this question, you know, whenever someone comes to me trying to think about how to develop an experimental program, everyone grasps onto the concept of, well, how big should my sample size be? Well, 30. Everybody uses 30, right? It's a very popular number. And I often get asked, why is it 30? Well, let's take a look at that. There is this curve called the power curve, and we look for the optimal use of resources in an experimentation program. Why? Very often our experimental programs are destructive, or at least contaminatory, meaning you test a sample, you can't reuse it for anything else because you contaminated it with the effects of the test that you're performing. We use very often, and it's quite popular, I get asked this all the time too, a binomial distribution with a C=0 acceptance as a test algorithm. Well, it's interesting when you look at these two, because there's a convergence. As the sample size goes up, the two graphs for those distributions, the geometric distribution shapes, they converge, they come together. And where that happens is actually right at, next click please, where the sample size is approximately 30. So if you ever wondered, where does 30 come from? This is where it comes from. It comes from the fact that your estimations, the binomial distribution and the normal distribution, usually based on a much larger population, they converge right at about n=30. And that gives you an optimal use of resources. Now, that said, is that the number you should use in your samples all the time, every time? No, it's not. We're going to look at and try to understand why that is so. And let's move forward and take a look. What you're trying to estimate here is the risk and the potential costs built into your experimental program. What are the pressures you have? Well, they're the little red devil on the right. Sometimes it's built in, hubris, right? Having an aggressive confidence that your test is going to work out just perfectly fine. Maybe you've got some, cost pressure from your CFO. You got to keep your experimental costs down because it's an expensive device, and I'll bet it is. And there's always pressure to go fast. That's pretty common in any company to try and get a product to market quickly. So you have those pressures driving you. What I want to encourage the audience to think about is what's really on the right. Understand differential costs of experimentation, right? Think about how much it tests to cost each additional unit. We're going to look at that in just a second and why that's important. Make sure you think about the costs of each kind of error you can make, a producer's mistake or a consumer, or in this case, a patient error, right? Think about progressive experimentation, and we're going to talk through that a little bit using double sampling plans here in just a minute. And I encourage everyone out there listening to think about what I call high veto power testing. Don't necessarily start your testing program with those items that are easy to test, right? Sometimes that's fruitful, but in more cases, it's actually more important to test those things in your device that are high veto power and could smash the whole program. That's where you start. It's a high veto power experimentation because it brings the whole program to a screeching halt. Don't be afraid of it. That's that drive out fear thing. Start there. All right, let's move on, because that brings us back to this battle for perfect information. All right, let's say you've gone ahead and picked an experimental algorithm. For sake of argument, we'll say it's a binomial distribution. We're going to sample without replacement. We'll set up a C=0 type of acceptance. And you had a failure. What now? Now is when you really want perfect information, isn't it? Well, let's go back to thinking about the reason I mentioned understanding your costs. What's an additional sample going to cost you? Is it cheap or is it really expensive? Just as important, what's your delay time? How hard is it to repeat your experimentation? Is it inexpensive to do, or is it really expensive multi-site type of trial stuff, right? Those are important elements to think through because they're all going to play into understanding now that you've had a failure in your experimentation. You need to understand the operational characteristic curve of your universe of parts or assemblies or devices that you're going to take and test, and maybe augment the sample size with more. All of that's necessary to understand to balance your risk with data so that you can improve your patient safety. All right, remember, you've already had a failure, okay? So let's look at this OC curve thing, right? I don't want to try and demystify it for everybody very quickly. This curve shows you where your confidence and power are located in the curve. So we'll start looking there. Let's just see where things shake out. You've got a producer's risk and you've got a consumer's risk. Let's take a look. Okay, your consumer's risk is shown down here at the bottom. That's your patient, right? That's how we consider the quality of an overall lot. Okay, if we've got bad quality, that means our patients are going to see that. That's not great, right? At the top is where we have our producers or manufacturers risk. That's characterized by confidence. All of that relates to the quality of the lot or the lot's total percent defective, okay? The way we're going to look at that, and the way I want you to start thinking about it, is what's the probability point at which you would accept this really poor quality? That's the important key point to think about your patient's risk. So as we set up our programs, right, we're going to want to think through incremental experimentation, increasing that consumer's risk down there at the bottom. That's higher patient risk. And that's why you want to be very cognizant of it as you go through these experimental test programs. Okay, let's move forward. So we're going to talk about a double sampling approach because you've essentially boxed yourself into that situation. If you started out with that binomial, a C=0, and you had a failure, you're going to be asked to augment your sample most likely and figure out if you have sufficient quality in that lot to actually be able to release product. Probably a very common occurrence for many of you out there in the field, okay? So since you're starting with a defective, you have to think about the proportion of your testing. I'm not going to go into the math with it here. I'll be happy to take those questions offline or answer them after the presentation with you for those that are interested. But it's important to make sure that you keep your proportions or your sample augmented sample size roughly the same or multiples of your original sample size. That's a symmetry question that relates to the probabilities, okay? But it's important to do that because if you do, the fidelity of the results coming out to you are meaningful. If you don't, there's an entrenched error in there that you have to watch out for. So keep it straightforward. Just keep the same augmentation sample size or a multiple thereof, okay? That's going to be why it's important to know the cost of each additional specimen, right? Usually there'll be pressure to keep it low. Depending on where you are on the OC curve, that's going to make a big difference. So let's move forward and take a look at what that means. We're going to start first by looking at it from the increase of sample size. Here's 4 different curves that show an N increasing incrementally from 25 up to 50. And what you can see here is if I take bad quality maximum limit as 10%, right? That's the lower axis. You see the vertical bar there. And my probability of acceptance there of roughly 30%, fairly high. And if I increase that sample size, I get even higher along the lower axis. That's the key thing to remember is, you know, as my N, I go to the left on the lower axis. As you see that sample size moving up, right, you see the curves are being pushed to the left. That's key. And we're going to look more at that here in just a second. Okay. So the recommendation here is to set up your samples with contingency in mind. Start with the standard plan. That's what everybody likes to do, which is the C=0 plan, but plan for that failure. Be ready for it, right? Have a clear instruction set for yourself to know that if it crops up, I'm not scrambling around to know what I need to do. I already understand it. I understand I need to keep my proportion roughly the same as my original sample or a multiple thereof, and I'm going to go ahead and augment depending on what the economics of my expansion allow, okay? Key questions to always ask yourself as you start thinking through this contingency, right? Are my samples random and representative? We hear that a lot. Are my processes, are they an operator and assembler training? Are they substantially equivalent to my manufactured setup, right? And is my supply chain stable? Why are these crucial? Because if they're not, what happens is your sample is less representative of truth and reality than you might want to accept. If you have any doubts about it, plan for what I showed down below. None of them are good, and that's why I encourage you to make sure all of these 3 bullet points up top are rock solid stable as you get into it. And here's why. Let's look at what this sample actually means next that we're originally going to take and that we'll augment with. The sample is not representative of your complete universe. If it's a device that you've just started producing, well, if you're successful, and we all plan for that, it's going to be around for decades, meaning that the universe of parts that will get created out of it is open-ended. Therefore, the gaps around your sample, the frame of which you're taking to do these tests, is incomplete. That's okay, but you have to accept it and own it. That's why the stability of those processes, people, and parts is so absolutely crucial. Okay, let's keep going. How does it affect our confidence and our power, or our consumers' risk and our producers' risk? Here's where we're going to look at it in some detail, just to give you guys a flavor for it. Notice that what I've shown you here is the C values as you increase. Remember, we already had a failure, so we already got C=1. We're now looking at C=2. But as I increase them, right, the producer's risk is shown down in the lower corner, right, with the little red box. Okay, let's zoom in on that and see what happens. Go ahead and let's click in and we'll see if we accept again a 10% LTPD, total percent defective, that's our patient risk, that ends up giving us a probability of about 12%. Okay, so this is for a sample size of, a lot size of 500, sample size of 35, and we set that LTPD at about 10%. That may be high for some of you out there. You may say, oh, I would never do that for my patient risk. But it's just a starting point to give you an idea of how we can balance this. Now, let's say that we have to increase our sample size, as we've talked about. Keep going, and let's see what happens. Oop, let's back it up just a little bit. I think we missed a click there. Okay, what you'll see, yeah, that's right. What you'll see is that if we increase that sample size and allow an additional failure, right? Our original curve is shown in the middle. Our increased sample size is the red curve showed on the left. If we get no additional failures, that's a lucky situation. We can't plan for it. If we allow for one additional failure, above and beyond the one we started out with, we end up on the left, and that shows us that we have somewhere between, type's a little small here, I think it's about 17 and 18% lot total percent defective for the same probability of acceptance. That's the key bookends to think about your patient risk. It's going to go up, but you need to know by how much, okay? And that's important because, let's move on to the next slide. Let's look at what happens now when we double the sample size. Okay, so we've doubled it. We're going to take a multiple of 2 over the original sample size. We'll keep this 5% lot total percent defective. That probably feels more comfortable to those of you in the device world. That gives us the same 12% of probability of acceptance. That's where our original curve is. It's in the middle. With one additional sample, we end up around 14%. Okay. The curve on the left, remember, is if we don't have a single additional failure in the augmented sample. Could happen, but we want to be prepared for if it doesn't work out that way favorably. What's the bookends of our risk? 5% to 14% bad quality in our outgoing product. Okay, that's the key. Understanding the bookends so that you can balance your risk with patient safety. Okay, let's go down. Let's keep on going here. The key takeaway, and it's a lot to cover in such a short presentation, is that your consumer's risk is your patient's risk, right? Augment your data set. If you're lucky enough not to have a second failure, you've actually got pretty reasonable product quality that you can balance that risk with. If you have to accept a second failure, at least you know how much additional patient risk you're taking on, okay? That's the key. And all of that's combined with your cost and time. Also equally important is that your sampling plans plan for more than just success. It's a pretty rare thing that industrial experimentation succeeds out the gate the first time. And that's why I encourage all of the folks that I work with to think about that potential failure and plan for it. It's very, very important. Okay, thank you so much for listening. We'll take questions shortly.
Thank you so much, Roy. Taking risk out of risk. I like it. So great presentation. I think we're going to have some good questions to come up with for you as well. As a reminder, everyone, you can download your slides through the download portion of your control panel. And then also don't forget to submit any questions that you have for the panel after these sessions. Our last speaker today is Andrew Thomas, and he is with WESTPAK. Andrew, it was tough getting some dirt on you. Just saying. Nora Crivello, President and CEO of WESTPAK, had nothing. She had nothing on you. She said you're squeaky clean. I'm guessing either she's not with the CIA or you're really good at hiding things. I'm not sure which. Since I couldn't scrounge anything up on you, I guess I'm going to have to play the Cowboys card on you. And while I won't hold it against you, I'm wondering how you came to be a Cowboys fan. So that's the question to ask Andrew the next time you see him in person. And just so you know, I know Nora won't hold it against you either. She told me she thinks the world of you. With that, Andrew, please share the information that you've got. And after Andrew's session, we'll be hosting the live question and answer panel. Thank you.
Awesome. Well, thank you for the introduction, Jen. Hello, everyone. My name is Andrew Thomas, and today I'll be discussing validated testing for medical device packaging. The aim of this presentation will be to touch on the process for validating test methods with a focus on medical device package testing. The push for validated testing has recently come from regulatory bodies in the United States and Europe. Specifically, there have been revised regulations in Europe that are tasked with improving medical patient safety. The aim of these revised regulations is to reduce infection risk due to microbial contamination of medical devices. In the next 15 minutes or so, we'll discuss general terminology, touch on regulatory requirements for method validation, discuss test method validation, or TMV for short. Then we'll go into examples of specific test methods, as well as walk through a TMV performed at WESTPAK. Before we dive into the regulations, I think it'd be beneficial to briefly discuss what test method validation it actually is. In one sentence, TMV can be described as the process of ensuring a test method is suitable for its intended use. This process involves establishing a method's performance characteristics and limitations by identifying influences that might change those characteristics. This is accomplished by evaluating the test method and is confirmed by testing. By going through this process, a TMV will provide an assurance that the test results will be accurate, reliable, and fit for its intended purpose. Performing validations on equipment and tests is not something new. However, the requirements for them to be performed on test methods has seen a recent push by several regulatory bodies. So let's dive into everyone's favorite type of bedtime reading, some government regulations. In the United States, the FDA, with the backing of the Code of Federal Regulations, or CFR for short, include requirements for process validation. Specifically, in the CFR Title 21, Section 820 defines what process validation is. The FDA also recognizes consensus standards ISO 11607, Parts 1 and 2, titled Packaging for Terminally Sterilized Medical Devices. Part 1 covers the requirements for materials, sterile barrier systems, and package systems. Part 1 of the standard is the main focus of this presentation. Part 2 addresses the validation requirements for forming, sealing, and assembly processes from a manufacturer viewpoint. So let's move on. Let's take a look at what the regulations look like directly from the CFR. I won't get into the exact details of this giant wall of text, but from Section 820, you can find a few of the requirements for process validation. Notice how many times the word validated or some form of the word validate appears. Each of these bullet points addresses different aspects of validation that must be met in order to meet these regulations and requirements. In Europe, requirements for validation are driven by the European Union's Medical Device Regulation, or MDR. The MDR was approved by the EU back in April of 2017. Its objectives include improving medical device quality, reliability, and safety. The MDR was originally planned to go live in May of 2020. However, like many things in life, the pandemic forced a one-year delay. The MDR is currently set to go live of May of this year and will replace the EU's current Medical Device Directive, or MDD. In Annex 1, Chapter 2 of the MDR, it states that devices labeled as sterile shall be processed, manufactured, packaged, and sterilized by means of appropriate validated methods. Requirements for validation, validated test methods, and validated testing are all key terms used in the MDR. It's funny, you'll notice that although this requirement may just be one small sentence, notice again how the word validated is key to understanding the requirement and scope of work that must be accomplished to show that these requirements are met. Now switching over to ISO 11607, you'll see similar statements and requirements. So let's briefly discuss ISO 11607 Part 1 and the latest 2019 revision. I'm told the writers of 11607 are doing their best to comply with the MDR's requirements. So as the MDR changes, ISO 11607 will follow. So you can expect new revisions to be forthcoming as these changes are made. So for Section 4.4, it calls out the requirement for test method validation. This section details some of the parameters and justifications required by TMV. This section states that a rationale or justification must be made and documented while selecting test methods. It also calls out for the requirement to establish and provide reasoning for determining the acceptance criteria of these test methods. Lastly, you'll notice our favorite word, validated, being used once again. In this instance, the requirement for validated testing falls directly to the laboratory, and it's pretty much the reason why I'm talking to you today. Section 4.4 and its subsections can be used as a basis for performing TMVs. So let's move on to subsection 4.4 or 4.4.4 of this section. It dives into the actual requirements of what a TMV shall provide. This section defines more clearly the specific requirements a TMV shall be able to prove. The three key items that a validated test method must prove are that it must have a determination of test method repeatability, determination of test method reproducibility, and an establishment of test method sensitivity for integrity test, which can be considered the precision and bias. Moving on to Section 9 of ISO 11607, it defines the requirement for package system validation. You'll notice that if the sterile barrier being tested first conforms with validation requirements per ISO 11607 Part 2 for forming, sealing, and assembly processes and meets the requirements for performance and stability testing, the package shall be considered validated. A key point to understand is that a package validation is a combination of a full suite of test methods. Each of these test methods must then be individually validated. Section 9.3 addresses any changes that can affect the status of a validated package system. The key takeaway from this section is that if any of these examples occur, they may require the validated package system to go through revalidation. All right, so next slide. Hopefully you're still with me. I promise that this is the last section on regulations. Annex B of 11607 is a list of standard test methods, guides, and procedures that can be used to demonstrate conformity. This section of the standard is multiple pages long and contains a wide range of test methods. As a third-party lab for medical devices, some of the more common test methods performed and requested by our clients are included on this slide. The tests listed here are a combination of distribution, also known as shipping simulation, as well as sterile barrier tests. We'll get into these a bit later. I know regulations are not the most exciting thing to discuss, but it's important to have a background as to why processes and test methods must be validated. The goal and purpose of these regulations always comes down to patient safety. All right, so moving on. So now for what I think is the fun part. So we've touched on the requirements, but how's this actually done and what does a TMV look like? Since we work with many clients that must meet these regulatory requirements for validated tests, the requirements for the test themselves in a way gets passed to us and is then verified by our clients. There are many ways to complete a TMV, and the approach we took is here. First, we dove into the requirements and regulations as we discussed in the previous slides. The next two steps, evaluation and defining the test method validation parameters, are essential to completing a validation. The bulk of the time and the effort will be spent during the first three steps, and if done properly, it'll make the remaining process much easier. So let's dive into evaluation. During this step, we looked at all of the test offerings we provide and determined which methods required validation for customers that are subjective to regulatory requirements. From a manufacturer perspective, they would need to determine which of these tests are required to perform a whole package system validation. This may or may not include all the tests that we at WESTPAK have validated. The key here is to determine what is appropriate for your intended purpose. For example, requirements of a device that is sterile versus non-sterile will require a different set of tests for validation. Moving on, so once the methods and intended purposes are identified, we then went into defining the test method parameters. The parameters set the framework for the execution of the TMV and address the intended purpose of the test method. A key factor in determining the parameters is defining the acceptance criteria and what a pass or fail result will be. Now, I want to touch on the different characteristics of an acceptance criteria for test method validation compared to the acceptance criteria of a package system validation because their purpose is very different. The acceptance criteria of a test method validation will be dependent on if the test method is qualitative or quantitative. And at this step, we're still talking about the parameters, so you can ignore this one for now. Perfect. For qualitative tests, like gross leak testing, the data that is generated is an observation based on defined characteristics, like a breach in the sterile barrier. The acceptance criteria for quantitative tests, on the other hand, like seal strength testing, will depend on a data set that is produced after completing the test and must fall within a defined range. If the operator is able to identify the known defects for qualitative tests and produce a data set that falls within the acceptance range for quantitative tests, this will then provide evidence that the test method is repeatable and reproducible. Now, there's a major difference for the acceptance criteria of a package system validation. The purpose of a package system validation is to show that your production methods produce safe and reliable products with adequate packaging that will arrive safely and remain functional after transit. The acceptance criteria for a package validation will typically require as little defects as possible based on an acceptable quality limit. Defects, however, do occur fairly often during the package validation phase. From our experience, it's not uncommon to see a package system go through multiple validation attempts. By clearly defining the parameters and completing test method validations, when these defects do occur, you can gain more confidence in the test results due to the level of evaluation that went into the test method validations. You'd also be able to be more confident in the results of a retest of a failed validation because you can be confident that they went through the same exact process and test sequence. Now, moving on to the final steps of the process. After the groundwork is laid by completing the first three steps, the remaining steps are relatively straightforward. After all of our parameters are defined, we create a plan for validation. This step involves creating any work instructions to align the consensus standards with your equipment. It also involves ordering any supplies that will be prepared for the test trials. At this point, we have several pieces of documentation that we need to ensure that the person performing the test, the operator, is trained and capable in. This will ensure the test method validation is performed and the data is recorded in the manner that we actually plan for. The execution is simply performing the test according to the test plan we defined in the previous steps. After all the data is generated, we review the results to ensure that they are what we expected. For example, for qualitative tests, this would involve ensuring that the defects were found and that no false defects were recorded. All of this is then wrapped together in a final test report. And that's it. I hope that this sheds some light on the process for completing TMVs, so let's move on to discussing a few of the actual test methods. There are many test methods that can be involved in a medical device package validation, which can be categorized into two main groups. The first is distribution testing, which many people refer to as the shake, rattle, and roll type of testing. Distribution testing will simulate hazards seen in the shipping environment. This can be considered the time a package system travels from the manufacturer to the end user or patient. The type of hazards typically seen during this process are inputs like drop, vibration from vehicles, as well as compression due to stacking. The second group of test types are sterile barrier tests. As you know from Dan's presentation, the majority of medical devices must be sterilized in order to be used to prevent patient infection. Sterile barrier testing will ensure that the sterile barrier remains intact and the device is able to be used without risk of infection. These tests may include gross leak testing, seal strength, as well as visual inspection. So moving on, let's briefly walk through gross leak detection testing and what a TMV actually looks like. Consensus standard ASTM F2096 details gross leak testing. It's a sterile barrier test that involves submerging a sample beneath the water, pressurizing it, and then looking for any breaches that would compromise the sterile barrier. To complete a TMV for this, we define the parameters and the acceptance criteria. We then ensure that the work instructions met the intended purpose of the requirements and of the test standard. The work instructions were also reviewed to ensure that detailed step-by-step instructions were included for WESTPAK's in-house equipment. After the operator's training was verified, they then followed these instructions and conducted multiple trial runs that included identifying a few different failure types. Upon review, the data that was generated showed evidence that the method provided repeatable and reproducible results. This showed that the test method was capable of identifying defects while meeting the acceptance criteria of the TMV and of the standard. So after this is complete, you may ask yourself, what does a final TMV actually look like? Well, here's an image of several completed TMVs at WESTPAK. Not pictured here is all the many hours that went into actually completing this process. Each of these binders is one TMV. I have a lot of respect for all you quality engineers and regulatory experts out there because I know this is not an easy process. These binders are considered complete packets that include work instructions, training logs, data recording forms, any raw data, as well as supporting documentation like calibration and maintenance records for equipment. All of this information is then presented in a final summary report that can be a standalone document, but it's also provided in the binders as reference. I know this may all seem a bit excessive, but when it comes down to it, validated test methods are the basis for improving patient safety. By completing these method validations, the manufacturer, along with the regulatory bodies, can be confident in the safety and reliability of their packaging to protect their products and be able to be safely used by their patients. For the last few slides, let's move on. If you would like some additional reading resources or just naturally curious about test method validation, here are a few references. If you want detailed information into TMVs, I highly suggest the first resource, the Medical Device Validation Handbook. It's a great resource with a ton of valuable information. And in case you're hearing about WESTPAK for the first time today, we are 100% employee owned and an independent test lab. We've been in the testing industry for 35 years with experience in the industries including medical device, pharmaceutical, electronics, and consumer goods. We specialize in a wide range of tests and have accreditations to ISO 17025 and ISTA. And lastly, if you would like any further information or would like to get in touch, please reference this slide. My contact information is here as well. Thank you for your time and thank you for listening, and hope you learned a little bit about TMVs. And from here, I'll pass it back to Jen. Thanks.
Thanks for great conversation and great discussions, Dan, Roy, and Andrew. If you guys all want to join back in with your cameras, that would be great. Being the pun lover that I am, I have to say that based on the data, you took a risk to provide information for the people in the industry on things they can do to improve patient safety. So I'm looking forward to our panel discussion to wrap up today. I'd also like to thank our attendees for spending time today to join in the conversation about data, risk, and patient safety. I'm personally hoping that if you didn't have a passion for this before our webinar today, that it's lit a fire in your belly to continue to improve patient safety. I'd like to now open formal question and answer period with our panel. If you have any questions, please don't forget to submit them on the question section in the control panel, and we'll continue the discussion here. So looks like you guys have all gotten over. I'm going to actually move my screen where I can see you because it's on my other screen right now, and I want to be looking what appears to be looking at you. Sorry about the noise in the background. My kids are finishing up lunch here. Hi guys, nice, great job. How do you feel? Did you like doing that?
Yeah, it was great.
Fabulous.
Yeah, it's great. Yay.
Let's do it at the beach next time.
Yeah, right.
Well, we were supposed to do it on site at WESTPAK with beverages, so adult beverages. Okay, so our first question that came in today, Dan, is kind of directed probably mostly at you, although I think Roy and even possibly Andrew might have something to add for it. So the gentleman that asked the question says that he's a former dentist and he's helping market a low heat plasma with a smaller unit suited for the dentist office in North America. It's got a 4-minute speed and it's safe to sterilize cell phones. And masks have already been proven apparently. Do you have any comments or help and availability for Tyvek bags for this unique process?
Yeah, it definitely sounds like a unique process. I know non-thermal plasma, generates electrons and some heavy particles. I haven't worked with it directly, but if you look at Tyvek, it's used with sterilization, which is hydrogen peroxide, which generates a plasma field. It's been used in that for years and years. Also used with enormous doses of gamma radiation, as well as E-beam, all the way up to 100 kilograms, and show functionality. So using that process, like anything, you would have to validate the process. There's not a consensus standard for that type of method yet. So you would use something like ISO 14937, which is a kind of a sterilization validation standard for items that there are not standards for, like 11135 is for ethylene oxide and so on. And you would obviously validate whatever packaging you're using with ISO 11607. So, as long as you validate it, you just need to show functionality. It should work fine, I would imagine, based on the other applications it's used in. So I don't see a problem using that. You can contact us. We have a network of authorized converters that can help with the application for that. Sometimes the films that you use have to be able to withstand that. So it's not just the Tyvek, you have to look at the film side as well. But we can help with that. So definitely can connect with you offline to find sources for that.
Yeah, a couple of comments, Dan. I think you're exactly right. I would say if it's non-thermal plasma, be careful when talking about devices that are software-driven electromedical. Direction of plasma, location, and intensity are key factors, particularly if your devices have non-volatile memory on board and require calibration. You could erase memory very easily. So a few precautions that you'll want to look at in terms of system design and approach. We work at Eagle, we're vertically integrated with hydrogen peroxide gas plasma. So we're quite familiar with the use of plasma in the sterilization process. You do have to be careful with it. The directional nature of it is really important. So depending on what it's intended to be used for, if it's just for simple mechanical devices, dental instruments and so forth, there's very little danger. But if you're talking about the software-driven electromedical segment of the marketplace, which is just exploding right now, I think you need to watch out for that.
Thanks, Roy. Andrew, did you have anything that you wanted to add, or Dan?
Yeah, I would just add just a quick tip. Think about your packaging early and test often, because we see a lot of people that go and think about that packaging at the end of the process, which they're ready to go to market at that point, and it's a rush. And I would just say think about your packaging and test early and often. So my tip.
Excellent. That's never a bad tip, having come from the packaging engineering side. It's a very good tip, because just when you think you know packaging, it's going to bite you in the butt and tell you don't know anything.
Yeah.
Dan, did you have anything to add or no? You good?
No, I think that's good. It's one of those things, there's a lot of unique different types of sterilization like this one that have really good applications. Obviously, that's going to be more of a smaller scale type thing. You're not talking 28 to 30 pallets at a time, but definitely has a niche. And like a lot of the other methods we mentioned, there's a lot of good options for some devices that can move to some of these other ones. But you really have to look at the factors about scalability, the ability to penetrate and get the necessary sterility assurance level, and material compatibility.
Okay, so our next question, with the EPA having high numbers, where did they get their numbers? Dan, that's probably mostly directed at you, because you've probably looked mostly into this one.
Yeah, I mean, the difference between the two models, it sounds like that's where that question is going. They both, that alternative proposal that the state of Texas is looking at, as well as EPA, they looked at the same NIOSH study of these workers that actually worked in the health, in these sterilization facilities. It's really comes down to the model that they use for the assessment. And generally the assessment, the levels that they're looking at is 1 in 10,000 up to 1 in a million excess risk of cancer. And so the models that they used, from what they say, I'm not a statistician, so I can give you the website for it. It's tceq.texas.gov. And if you just Google TCEQ ethylene oxide, they'll come up with their page, and they do a good job of explaining the differences, where they got it. And they talk about the EPA using a kind of an unconventional supralinear dose response model, where they use more of a different type of model that's the Cox proportional hazards model. So I'm by no means an expert to get into either one of those, but they do a good job of talking about that. So it's really how they did the assessment of the data and modeled it. And from what it sounds like, from everything I've seen, the Texas model, actually the data fits better, and it actually predicts the actual observed cases of cancer way better than the EPA, which went way over predicted it. Hopefully that helps. But you can Google that, and it'll go through. They have a long list of questions on how they came up with that model.
I think the one that always blows me away, Dan, is the fact that humans themselves put off more ethylene oxide than most of the sterilizers out there. Like, that just,
yeah,
it says so much right there, right? Okay, so the next question is, X-rays are currently being used for medical device sterilization at the Synergy Health Facility in Daniken, Switzerland by Synergy. You know what?
Yeah, it's more of a statement.
Yeah,
there's a couple facilities that are using X-ray now, so it's not brand new. Like I said, it's been around for a long time, just not as widely adopted because it's maybe not as efficient as E-beam is. So you in real simplified terms, you have an E-beam unit that you kind of convert into making X-rays, but that conversion process can be like 16%. And so it's not very efficient. Of all those electrons you produce, you're only getting 16% X-ray. So then, that's probably why it hadn't been more widely adopted. But it's got some great advantages. It actually penetrates even better than gamma, and so but less material issues than more similar to E-beam. So it's kind of the best of both worlds as far as between gamma and E-beam. So it can penetrate even dense products and be used on a large scale. So I think you'll see a lot more of that.
Yeah, I'll make one comment, Dan, about that, the I guess awareness level about the various modalities of sterilization. I'd encourage the audience to not fall into what I call the rut of the predicate problem. Realize that right now, under FDA emergency use authorization, thanks to this lovely pandemic we're all experiencing, they're much more open to accepting applications that use sterilization modalities that are generally accepted. X-ray is one of them, E-beam is one of them, just like gamma, EO, and hydrogen peroxide are all one of them. Take a look at all of them, because it's all about getting the best bang for the buck and the highest degree of sterilization reliability for your process. It's important. If you simply go with what was done before, you may be doing yourself a disservice.
Thanks, Roy. So Roy, you didn't actually have a lot of questions, but I have a couple of theories behind that. And one of them is kind of summed up in a statement that's made. It says, there's no question, but this guy, Roy, is really awesome and engaging.
Oh, gosh.
Actually, some great leadership shares as well. So there's some kudos to you. I also had a text from an attendee indicating that she's hoping there's a recording of this session because she wants to let your message distill a little bit. And my response back to her was, I'm horrible at statistics, so I'm not even going to try. That said, though, two things. Yes, it is going to be recorded. It has been recorded and will be shared later, so that's great. Roy, well, kind of three things, I guess. Roy, you indicated in your presentation that you're open to having discussions with people about this,
Sure.
and I believe your contact information was in your slides, and if it wasn't, we can always get those people in contact with you. But third, if there was like a statistics for dummies, or if you have a specific example where the double sampling came into play, if you can dumb it down for at least me, to help me understand a little bit better.
Sure, be happy to put something like that together for everybody. I think it's helpful for people, that contingency plan when you start out, because the world just never goes how you want it to. We've all been there at 11 p.m. in the lab running a test and it fails, and now you can't sleep for the rest of the night because you got to figure out what you're going to do tomorrow.
Usually comes in on Friday afternoon, right? Just as about
Yes.
as you're
Been
going to
there,
go on
done
vacation.
that.
Yeah.
It's
Exactly.
never the fun news to deliver from the test lab side.
No, well, and when we were prepping for this, Andrew, I think you said, and you touched on it before when you talked about bring packaging in early, but you also commented about the number of iterations that people need to do sometimes because of that situation. So, clearly there's probably some design changes that could be made early on. People could contact you as part of the design process and say, hey, have you seen anything cause problems that we have in our device? But then you can also, be talking about this double sampling concept as well and help prevent that from happening. Nora's probably going to be yelling at me right now because it means less business for you guys, but ultimately you guys want the success of your customers as well as is your number one mission in life. So now, Andrew, I have some questions for you.
Sure.
The first one is a little bit tricky. Can you claim compliance to 11607-1 without a packaging validation that includes loose load, compression, and concentrated impacts?
So the specific test types will really come down to justification for your specific product. So there's not really a one size fits all, like it doesn't always include loose load, doesn't always include compression. So as long as you have the justification for why you did not include those or why you did include those, then you can complain, or then you can claim compliance. It just really comes down to justification for your specific application and why you chose the test you did. So that's what it really comes down to.
Exactly, that comprehension and understanding of your distribution modality out into the end user. Roy, do you have anything to add? Because I think that you've probably worked through packaging validations from an MDM type perspective.
Yeah, so the one thing I would say, dovetailing onto Andrew's statement about the justifications, don't be afraid to use them. They're very powerful. I often find that in the industrial experimentation community, people repeat experiments that are absolutely unnecessary, and simultaneously will overlook experiments that they think are not crucial, but actually are. That's all ties into that whole thing we talked about with producer and consumer's risk. So where you can use a justification, take advantage of it. It's time and money saved. Use the high veto power approach. I'm sure Andrew's seen tons of tests come through his lab where it's a yard sale in the drop test room, and all of a sudden the engineers are scrambling around trying to figure out, like, how do we fix that? And people think, oh, that's a bad thing. I'll put you back to the video where we started out, where Elon Musk blew up a $21.6 million dollar rocket and said it was a success. That's how you got to think about it. It's a high veto power test. He got what he needed out of it so that the next launch will not have that problem.
And if I could piggyback on that too, you could always split. You don't have to do the validation just as one go. You can always split it into individual tests. So if you think you're going to have a tricky issue with, say, drops, for example, you can always just do drop testing and then do that as its own separate thing before you even start the validation phase. So I would always say test individual tests that you think you might have an issue with before you actually dive into the full validation as well. It helps a lot.
Excellent, thank you. Next question is packaging related as well, and I think probably both of you guys that just answer that question can touch on this one as too. When talking about TMVs, sensitivity and the role it plays in your test method validation, can you talk about how you deal with that and its impact on your successful TMV?
Sure. For this one, it's a little bit more tricky because it really depends on the method itself and what you're actually doing. I'll probably follow up individually with this one, but Roy might have a better answer than.
Yeah, let me offer a little something here if I could, Andrew. So test method sensitivity really boils down to understanding the factors that influence your test outcomes, okay? So you gotta know all of the experimental factors that a test is intended to stress, right? So if you think of them like rubber bands, each factor is gonna get stretched when you put it through the test. And understanding how much it can stretch or how little it can stretch is essentially the sensitivity. It's really sensitive if you can only stretch it a tiny bit and it breaks. It's not very sensitive if you can stretch it a mile and it doesn't break. So understanding what factors have that kind of effect in your experiment, that's what sensitivity is all about.
Thanks, Roy. The next one I think will be quick, and hopefully we can answer it quickly because we've got a bunch of great questions still to answer, but we're really quickly running out of time. How do we choose between ASTM and ISTA?
Ooh, Andrew, yes, I'd like to.
Again, it's justification. We see a lot of mix of both, but as long as you're consistent and have justification for why you choose one, ISTA actually references a lot of the ASTM standards. So we see mostly ASTM, but a lot of ISTA as well. So this is the basis of it.
They're both pretty robust systems.
Yeah, they're both really great. It just depends.
Yep. Next one is also pretty quick. Is a TMV needed for basically all test methods?
It depends on, again, what your purpose is. So if you are trying to be compliant with ISO 11607, then yes, all the tests under that umbrella need to be validated, go through this process. But if you're just want to have like, or just want to do an experiment on the side, then no.
So I'm going to add something onto that, not having worked at a test lab facility, but at an MDM before. If you're not performing the tests yourself and you're having a lab perform them, before you contract the lab, highly recommend that you ask the lab if they have their test methods validated. Because if they don't, then you have a big problem, right? Basically, to your point, Andrew, everything does need to have a TMV completed if it's going to be supporting 11607 justification.
And doesn't your lab accreditation, Andrew, doesn't that usually address that, right? The 170.
Yeah, we have 17025. It does.
There you go.
But it doesn't go into specific requirements like 11607 does for TMV. So there's no reference to TMV for 17025. That makes sense.
Okay, here's a comment for all of you guys, and there might be a question. Oh, the question has already been answered, but great webinar, a lot of information which they'd like to review again. Recording will be available just to answer that a second time. Let's see here, Dan, you made the case for a need for science-based solutions to the challenges of EO. Do you see this being successful when misinformation is so prevalent, like we saw in Willowbrook? And there's another one from earlier that kind of piggybacks onto that. Is, do you see any more closures to EO facilities coming up?
Nothing that I hear.
EU, right?
Yeah, nothing that is in the works that I've heard of. I mean, it's going to depend on these emission limits that come out. A lot of it, if you've looked at what's happened so far, it's been individual states that have taken action. So and the technology is there to be able to reduce these emissions, and you know, we do have the technology to do that. You know, whether they will take a more science-based approach, we hope. There's groups like EOSA, which is the Ethylene Oxide Sterilization Association, that is working with, you know, lobbying EPA. There's AdvaMed, other industry groups that are trying to, you know, present the good science and look at the balance of the risk versus the benefit and what would be catastrophic catastrophic if this method went away. So, you know, if I had a new product and was looking at doing EO sterilization, I don't necessarily think I would be afraid of going forward with that, because you know, there's just nothing that can replace it. It just can't go away. There's alternate methods, but you know, due to what we've talked about, you know, it's going to be a long time before ethylene oxide can be replaced. Something else will have to come up that doesn't exist right now.
Hey Dan, I got a quick question for you on that vein. What is the industry sort of saying is the timeline for facility expansions to come online? Is it kind of like a 1 year, 2 year thing? Are we talking like 5 to 7 years? What is the industry saying?
For expansions for EO facilities, or
yeah,
are you talking? Yeah, I mean, I think right now, just because of the uncertain regulatory landscape, you don't see a lot of people installing new chambers or building new EO facilities, you know, probably as a business decision yet. So I think I know of one sterilizer that's actually installing new chambers right now, but you know, it just remains, everybody's sort of taking a wait and see approach to see what happens, which hopefully we'll know in the next couple months of where that is. But you know, you just look at the huge, and 50% of all sterilized medical devices. I mean, that's, when you look at the impact that could have, you know, the choice is pretty clear there. There's ways we can deal with it. There's technology that we can work with it very safely. And, you know, already the technology in these facilities is very good.
So moral of the story is EO's not going anywhere. If you have something validated in EO, don't necessarily think that you have to switch it, but you should probably be cognizant if there are other modalities available that would work with your product in the event that something does happen. Would that be a fair assessment?
Yeah, I think so. I think a lot of manufacturers are looking at their devices and looking at options. You know, in the event something happens, you know, it'd be probably prudent to say, you know, can these go a different way? You know, you're not seeing any wholesale changes of companies moving all their products out of it. There just really isn't a way to do that when you're talking on such a large scale. Even the regulatory requirements for switching from one modality to the other, you know, I've had some companies say that it can be years to do all the, you know, you got to do biocompatibility work, and you have to do all this data that you have to get to show that whatever method you're looking at with that type of with your actual product, that it can be safe and effective. And so there's a lot that goes on to switching modalities. It's not something you can do in a week, for sure. You're talking years in most cases.
Well, you guys, we are coming to the close of our fun today. So I'm going to close the questions at this point. Great panel. Thank you very much, you guys. Maybe we'll try to get the gang together again and finish answering some of these questions offline, and then share on LinkedIn channels a little bit later. So as previously mentioned, slides can be downloaded from the handout section in your control panel. A recording of the session will also be made available soon. Thank you again for your time today. If you'd like a follow-up, or there are others on your team that weren't able to make it today, please let us know. Keep an eye on your inbox for upcoming emails from our team, and we look forward to talking with you again soon. DuPont has a global team of specialists ready to help you in your medical and pharmaceutical packaging needs. And you know, we'd always be able to help you get in touch with both Eagle Medical as well as WESTPAK. Any which way, I know that all of us would love to connect with you further. So visit us at medicalpackaging.dupont.com. Follow us on LinkedIn. Follow the other companies on LinkedIn as well. And we'll post all of our upcoming events there. This concludes today's session. Have a great day. Take care and be safe out there. Thank you. Bye.
Thanks, all.